bit about my journey. I don't have any take-home messages. I'm not very good at giving advice, but I am a storyteller, so I am going to share my story and perhaps you will take something out of it. Yesterday, we were talking about when to start talking to children about science. Which grade? This is grade 3. I had just moved to a new school. Can you hear me now? I had just moved to a new school. It was an English medium school. My parents really wanted me to go to an English-speaking school. Both my parents are microbiologists. The school was just starting their first science fair because both my parents were microbiologists. The teachers came up to my parents and said, "We are making your daughter a group leader. She has to come up with a science project." My parents, this was also their first science fair ever, came home. We had a whole dinner table conversation on what to do for science fair. And we decided on a project called blood grouping. The idea was all my group members will learn how to do blood grouping, the science behind it. We will teach everyone at the science fair how to do blood grouping, how it works, and everyone who comes to visit our booth, we will actually take blood and group their blood and give them a report. This is early 90s, so most people did not know their blood group. That's the first time I learned that biology and public health go hand in hand. Every teacher, every parent came to school, went home with their blood group. So that was my introduction to science. A lot happened after that. After grade 3, I made many friends, but in grade 11, I made a friend who became my best friend, Nabila, and in grade 11 in Dhaka, Bangladesh, I was like, there's no way I can ever live without her. Half of her family lived in Canada, and she was applying to go to Canada, so I stole my dad's credit card, which turned out to be his friend's credit card. Not a good idea, don't do that. To apply to 4 schools in Canada. I got into all 4. University of Toronto gave me a scholarship. My parents did not know, this is before the time of email, so letters got sent to our address. My mom happened to open the letter. It was not nice, but we are friends now, all good. And then I got a scholarship. They were like, people go to the US, where is this Canada? But I did my bachelor's in biochemistry at the University of Toronto. I was an excellent student. During my 4th year of biochemistry bachelor's, I actually started working in a lab where I knew I wanted to do my PhD. I did so well that directly after my bachelor's, I immediately started my PhD project. And I say started because I was doing so well in the lab, I wanted to make a record of how fast you can get a PhD at the University of Toronto. By 18 months, I passed my qualifying exam with flying colors, ready to finish my PhD in maybe 3 years. But then I was diagnosed with cancer. It turns out, I don't know what happened. Damn it, you get all my story before I tell you. But anyways, when I was doing this, preparing for my qualifying exam, it turns out a tumor, 6.4 centimeter, was growing on my neck without me realizing it. So that's the size of a tennis ball. So right after I qualified for my exam, it was all, hospital visits, surgeries, therapies. So my plan of being the fastest PhD student did not work out. This is right after I came back, so after a year or so I started coming back to the lab. You see me wearing a scarf because I was very conscious about all the scars I had. So cancer did not only leave me with a lot of physical scars, but also a lot of mental health problems that I had to deal with. At that time, I never thought I would come back to my PhD, but I did because of the amazing supportive family and PhD group that I have. And finally, after 7 years, I graduated from my PhD program. After that, I started a postdoc at Hospital for Sick Children in Canada. It's the largest pediatric hospital in the country. It was on translational medicine, so my PhD was on finding novel antimicrobials for Pseudomonas aeruginosa. I wanted more translations, so I joined that department, did not really like it. Within 4 months, I actually left that that lab because it was a lab in Canada trying to work on the problem of malnutrition in Africa. Being an activist, somehow it did feel right. If I wanted to work on African problem, I had to be in Africa. I couldn't be in a lab in Toronto. So then I went to Stanford and did my postdoc on epidemiology. That's when I learned R, that's when I learned coding, modeling for public health problems. While all of this happened, I also married. This is Dr. Yogesh Huda, also a very, very basic scientist. And I must say, to this crowd, he's a Haryanvi Jat. Haryanvi Jats can also be scientists. Not only did we marry, we also adopted our first cat, Ava Fontaine. If you watched Nicolas Cage's Lord of War, there's a character, Ava Fontaine. Yogesh was a big fan. So we named Ava Fontaine, our cat Ava Fontaine. So during all of this, Toronto really became home. Toronto was comfort. Toronto gave me free healthcare, which continues to be very, very important for me. While I was at peace with my personal life, with everything in Toronto, there was something that also bugged me. I didn't know how, my work in Toronto will ever reach my people in Bangladesh or South Asia. Also, I realized, cancer defined a lot of who I am. I realized if after PhD people have 40, 45 years to work, I maybe have 15, 20. So I wanted to use my time as effectively as possible. And my professional interest really lied in South Asia, in Bangladesh, specifically in Bangladesh in this organization called the Child Health Research Foundation. This is an organization, it's a non-profit organization working in Bangladesh since the 80s, and they work towards improving child health by working in the lab and ensuring that all the laboratory data are actually translated such that policymakers understand and right policy decisions can be made for the country. This hospital has done a great job. Our headquarters are in Dhaka, Bangladesh, and we have a network of 4 different pediatric hospitals. And in 3 of these hospitals, we actually run the diagnostic labs, and we run the surveillance in these hospitals. These are all now WHO sites. So really, this is this beautiful combination of laboratory work, clinicians, hospitals, samples, and data. So it seemed to be the perfect place to be if I wanted to work in Bangladesh. I specifically, the Child Health Research Foundation, was interested in meningitis. Meningitis is an infection of the brain. It's a severe infection. If we do not treat meningitis immediately with the right antibiotics, a lot of children die. And if there is not optimal treatment, actually more children than die. Most children are left with disability for their entire life. So even when I was in Canada, the 11, 12 years that I was in Canada, whenever I was healthy, I would actually come back to Bangladesh and work as a community health worker to understand what happens to a family when a child is disabled. And in our society, in South Asia, in a poor family, when one child is disabled, the siblings fall out of school, mother leaves her job, the father goes into debt. So really the entire family becomes disabled, the community becomes disabled. So I was really interested in working on meningitis. When a child comes into a hospital with symptoms of meningitis, which is usually high fever, seizure, lethargy, what the clinicians do is do a spinal tap or a lumbar puncture. So they put a needle inside the spinal cord and take a little bit of the cerebrospinal fluid, or called CSF. CSF is sterile. It should not have any blood cells in it. However, if there is an infection in the cerebrospinal fluid, then white blood cells rush in to clear out that infection. So if we see white blood cells in the CSF, it's indicative of an infection. So over here, I am showing you data from 2015 to 2022. Our labs, which are part of the largest hospitals, as I said, has processed about 9,000 or 8,500 CSF samples. And what you see here is 40% of these CSF samples have high white blood cells, so more than 9 white blood cells, indicative of infections. All of these samples go to certain tests in every microbiology or biochemistry lab. And despite doing these tests, that includes culture, serology, PCR, what we find is that 80% of all meningitis cases remain idiopathic or remain mysterious. In other words, we have no idea what causes 80% of meningitis cases, and that's true across South Asia. And this is a problem because when you don't know what causes disease, you don't know what to do about it. My lab provides reports to clinicians, so 80% of the reports go as, meningitis, but we don't know what caused it. So clinicians have no idea how to treat it. We cannot work with policymakers to figure out what vaccines to introduce next. So our organization was the organization that worked with the government to introduce the HIM and the pneumococcal vaccine in the country, but we don't know what vaccine to introduce next. So when I was making this decision of moving to Bangladesh around 2015, I hypothesized that if we used a technology called unbiased RNA metagenomics, we will probably be able to answer some of these questions, solve some of these mysterious cases. So what is unbiased RNA metagenomics? At that time, it was still quite new, and pretty much nobody was working with CSF samples because it's a sample that's very hard to get. And even if you get the sample, they're usually not linked to clinical data. So unbiased RNA metagenomics, in this case, would be, you take the CSF sample, extract all of the RNA in it, and all of the RNA basically means all the human RNA and any pathogenic RNA. I chose to work with RNA because all organisms at some point of their life have RNA, not all organisms have DNA, for example, RNA viruses. Then you prepare a library for sequencing, you sequence the shit out of it to get a lot of data, and then you analyze that data, you build mathematical models using the right positive, negative controls, sequence basically everything, and then you figure out what are those reads from the pathogens that is a potential etiology or cause of disease. As I was hypothesizing it and reading a lot of literature, looking at all the other countries in South Asia, I realized this is not a Bangladesh-specific problem, right? This is a problem that many countries in Asia, Africa deal with, and it is also not a problem that I can solve by myself. So I definitely needed expert collaborators. So I was also, by then, I was also reading a lot about our colonial roots, how our health is still colonized, and how it's extremely important to decolonize health. So before I even started collaborations with international colleagues, there are 4 clauses that I put in. That's how I would start my conversations. You want to collaborate, but these are the things you need to remember. Any protocol that we use to solve these cases of meningitis has to be open access, so all across the world people should have access to the protocols. Any technologies that we use must be transferable to Bangladesh, because if it is not transferable to Bangladesh or South Asia, that technology has no meaning to me. And at least one Bangladeshi scientist from our organization has to be trained on the techniques, and at the end of the day, Bangladesh will own the data set. To me, this seemed the right things to do, but turns out it's not normal, and oftentimes South Asia, when we have international grants, we lose our ownership of data. So I had to be very, very particular about this. And hence I had a series of failures. So 2015 I submitted a grant to the Gates Foundation. 2016 it got funded. We hadn't signed the agreement, it got funded. It was a collaboration between our foundation and another institute in a different country in Asia. And while they did verbally say that they would maintain all of the, or they would agree to all of the clauses, it was very clear when we were signing the agreement that they would not share the protocols with us, and it would be in a completely different language, and they would not even help us translate the protocol. So while I left my husband, left more importantly my cat, to come back to Bangladesh thinking I would get this grant, I did not end up signing the agreement because I would not be able to live with that. I moved to Bangladesh to specifically set up this protocol. So that didn't work out. I worked with a collaborator in Canada. We tried 16S sequencing, which is different, but the collaborators were not collaborative. The results were not very exciting. And then I went to this typhoid conference because the years when I didn't have any grant, I was basically working on epidemiology. With this foundation, I went to a typhoid conference and we were walking by a lake. This professor from UK basically said, Senjuti, if you had to solve one problem in this world, what would it be? I said, of course, solve the problem of meningitis. He's like, that's no problem. Next week, he showed up with his postdoc in our lab in a suitcase full of reagents, and we tried to use nanopore and MinION technology to solve some of these, sequence some of these CSF samples, solve the problem. Unfortunately, the technology was just not ready enough then. My biggest problem working with all of these groups were they really had no expertise in working with CSF samples. So this is the beginning of 2016. By 2017, I'm frustrated. I'm extremely angry. I get invited to give a talk at the British Museum in London in November of 2017, and I give an angry talk. I'm always angry. I was extra angry when I was at the British Museum, and I gave a talk, and it was called The Dark Side of Meningitis, Why Investors Are Not Interested in Funding Mysterious Diseases. That attracted a lot of attention, so being angry helps. As Maha said, I immediately after that, I got a call from a senior program officer at the Gates Foundation, Dr. Farhad Imam, and he connected me to a professor in UCSF, also the co-president of Chan Zuckerberg BioHub, Joseph DeRisi, and we got on a phone call within a week. Farhad was in my lab. Within a couple of weeks, I basically was in San Francisco with 96 samples. I did not take the samples with me. They were shipped separately, dry ice. I did not get any money for it, but the offer was, you come, we will follow everything that you want. Joe DeRisi had developed this new analysis techniques, but he didn't have samples to try on. I had samples, but I didn't have the techniques. It was a perfect collaboration. And that became a project that, like a very, very successful project. I was in SF for 2 weeks, and not only I learned all the techniques, how they do the sequencing, but also we were able to look back at some of the meningitis cases in 2017 in Bangladesh. And for the first time in the world, we showed that chikungunya virus, a virus that's carried by mosquitoes, can actually cross the blood-brain barrier and cause meningitis. Right, so it of course needed a lot of work in addition to the sequencing, but that attracted a lot of attention. We were able to publish very quickly, but before we published anything, I got a call from Ed Young of The Atlantic, who was interested. That was my first journalist interview. Since then, I've done many interviews, but that was the first one, and my favorite science journalist. So we got covered by The Atlantic. That tracked, again, more attention, not only attention of scientists, but also attention of donors. That led to my real grant. I got my first grant to actually set up sequencing in Bangladesh. So that's a small sequencing machine right over there. That's an IISER 100, a global sequencer. It can be put in the back of a truck, taken anywhere, that can sequence. However, it produces really little data. I can sequence 1 bacteria at a time, or maybe 4 bacteria at a time, or 1 CSF samples. That's my group using the sequencing machine. So to this date, when we use our sequencing machines, we are like, break a coconut and then run it. And we've probably done, 70, 80 sequencing runs. I was invited by Bill Gates to speak during the UN General Assembly meeting to talk about advancing health and why we have to close this technology gap. Then I got my third grant, which was basically to build a whole genomic center. So first I got a $100,000 grant, then I got a $1 million grant to basically set up genomics in the country and start chipping away at understanding what causes diseases. We have built a Bangladesh pediatric observatory, so for all bacteria that come into our lab through the different hospital surveillance, we do whole genome sequencing, we do viral genome sequencing, and of course, we try to figure out what are causing these mysterious diseases, and have become very fast. Since then, everything has moved really, really, really fast. After that first project that worked out, we were the first to sequence SARS-CoV-2 in the country, part of the surveillance consortium. We have sequenced hundreds and hundreds of salmonella. We actually built the software that is needed to analyze genomes of Salmonella paratyphi A. It was published in Nature Comms, but I must say we also did not, like Andrew Lin was saying, did not publish this software first. We put up the tool on GitHub, people were using it, they were citing the tool from GitHub, we optimized the tool, and later we published it. We also found the basis of azithromycin resistance in salmonella. salmonella, first to show that in the world. We have sequenced 600 Klebsiella pneumoniae. All of that data is now being used to design vaccines for Klebsiella pneumoniae by different groups in the world, and really so much more. I have always worked with things without nucleus, but now we have even moved on to things with nucleus, if my slides ever move. So now we also do single-cell genomics, so we are trying to understand what are the cell types that we have in a nasopharynx. So this is hot off the press, we just generated our first single-cell data set last week. So there's really a lot of exciting science happening. These are all my donors. Over the last few years, I've been able to raise about $7 million US, and all my grants are international. We don't have a lot of funding, national funding in the country. So if you're complaining about national funding in the country, don't, right? Please be grateful. However, if you wanted to, talk to me about how to get international funding, then I can perhaps have a discussion. Depends on what you buy me. I've been covered by the Lancet for my work on equity. I've been named as one of the top 100 researchers in Asia. Bill Gates made a documentary about me and my work with my father, who works also on vaccine introductions. And I also received the highest civilian award for women from our Prime Minister. I was the youngest. But most important for me is that I work with a wonderful team. Our team is growing very fast, but it is just a wonderful team, and I think this is where Maha and I differ a lot. I do not care how they work when they come in. I am angry, but how they work is their lives, and they have their own teams, they get things done. I also have people who come, who leave, and that's okay. Each individual team figures that out, but together we just have a lot of fun, and we have a lot of game nights. Our labs are full of card games and table tennis, etc. But this is also the time. I might sound very successful, but I want to say I'm also extremely stressed. I've never been this stressed, and I've been through a lot of health issues, and my stress comes from the fact that the team has grown. All my funding is international. I write grants like no tomorrow. I was telling Raghu that I wrote 23 grants last year. I can write grants in my sleep. Not sustainable. So how do I figure out how to maintain this team and we can continue to do amazing science? I'm becoming, I have 5 more minutes. I'm becoming an entrepreneur. And I think that's very important for us to think about too. I see myself as a CEO of a startup. Others might not, but I think that's what I do. I run a startup. So now all the machines that we have set up with these grants, we are trying to use them for the country and also make profit. So I do now sequencing for the entire country. Academics who used to send their samples outside the country to sequence them, we sequence them in-house, provide very quick results. For next-gen sequencing, for Sanger sequencing, we also just inaugurated our genetic diagnostic lab, again using the same expertise, same machine, so using resources optimally, doing something for the country, also ensuring sustainability, and also how do we deal with the inequality and the prejudice. I'm a woman, I look younger than I really am. I am young, but I look younger. When I moved to Bangladesh, it was all about how did you leave your husband to come here? What is he eating? And then with my women colleagues, I talk more to their parents than them. So living in this equality, in this society of inequality and prejudice, it's very hard. It makes doing science very hard. So in 2022, we decided to make a change and we rolled out this program across the country called Building Scientists for Bangladesh. It has 3 different streams because we want to change one mind at a time. We want to change mindset, change a society. So in stream 1, we travel across the country setting up science camps. We take microscopes and petri dishes with us. We stay for 2 days. In stream 2, we bring people to science. So school students are invited to come to a research institute with their parents and stay for a few days, because it's not only students and girls' minds we are trying to change. We're changing. We have to change parents' minds because they make decisions for their kids, and for our stream 3 is basically skill development, where professionals can come in and train and use the machines that gets used once a week. So other 4 days are just open for anyone who wants to come use the machines. We have so far reached 10,600 school students across Bangladesh and trained 400 students on campus. And while it is called Building Scientists for Bangladesh, it has gained a lot of attention from the international community, and we are training students from the entire world, which I'm very proud of. So we are really showing what countries in South Asia are capable of. I'm almost at the end. There are a couple of things I did not cover. If you buy me coffee, you always have to buy me things. Thinking like an entrepreneur now. Buy me things. If you want to know how I hire into my team, we have very specific protocols for this. I can talk about that, how to let go from the team, as important when you have a big team. Happy to talk about that. How to accept failures, I don't know, but can vent with you. If you buy, and how to deal with a society. What does your husband eat? Have you separated? Why are you keeping girls after 7 p.m.? What are they doing? If you want to talk about that, you need to buy me something strong. However, in case you're interested, what happened to my husband and to my cat? They moved to Bangladesh. My husband, also a scientist, is setting up a basic biochemistry lab at our foundation. Ava Fontaine has moved with him. She is now an 11-year Canadian feminist, having a territorial battle with the 2-year Bangladeshi male chauvinist at the border. I'm going to leave you with this quote. Constantly ask yourself what we're doing for others, because that's what people will remember. People will not remember our publications or our impact factor. Thank you.
So much. That was really, really inspiring. Be the change you want to see. Real example. We'll have only one question. Yeah.
So, very good talk, and congratulations for the achievements. So, have you, in these patients, first, what is the age of these kids? Second, whether they are born normally or C-sections, and what are the microbes, especially bacteria, they are dominating?
Yeah.
Thank you.
Thank you so much for the question. So your question is the cohort of patients from where we are getting the CSF samples, their age group. So all our children are under 5 years of age. We are seeing a lot of infections in the first 28 days of life. As for what bacteria or pathogens are causing these infections, the younger the children are, the more gram-negative bacterial infections we see, so Klebsiella, Acinetobacter, Pseudomonas, etc. However, a lot of the other pathogens we are seeing are like mumps virus. So Bangladesh doesn't give MMR. We don't give measles, mumps, rubella vaccine. We only give measles and rubella vaccine. Mumps in 40% of the cases is neuroinvasive, which I learned recently. So we see a lot of mumps meningitis cases. Of course, I talked about chikungunya. We see a lot of dengue meningitis cases. Currently, we actually discovered new virus, a family of parvovirus that we're studying, also causes high rates of meningitis both in India and Bangladesh. Hope that answers the question.
Are they like regular born or C-section?
Oh, if there's a correlation between vaginal delivery and C-section, we haven't looked at that, but most of the deliveries, unfortunately, in Bangladesh is C-section now. We are moving towards that direction, which is a huge problem for the country. But we haven't done that analysis if babies with C-section are more likely to get infections.
Yeah, we'll continue the technical questions over lunch, but before the next speaker is setting their slides, I have a question like, with large team, how easy is mentoring for you?
I definitely cannot mentor everyone in the team. There are people in the team gradually, especially, I did show their pictures, we have about 100 community health workers now. Of course, I do not even know all of their names. But I think mentoring mentors is also very, very important. We have a group of 8 amazing people that I work with. We meet every day almost if I'm in the country. And if I'm not in the country once a week. We talk about all of their groups, their students, and the way the organization, or at least my teams are set up now, is that even if someone leaves that team, there will be no loss of skills. I think that's also very important to remember. One is mentorship, the other is accepting that some people will leave. And the best advice I got actually from Dr. Cherry Kang of CMC Vellore is let people leave but never lose skills. And I think that is my mentorship guidance for mentors that I mentor. Yeah.
Thank you so much. Thank you. Please give a big hand to Dr. Saha.